Bismillah ar-Rahman ar-Rahim. Assalamu alaikum jami'an. This is the third lecture about transcription. You remember that we talked about the transcription in prokaryotes and eukaryotes in the previous two lectures. Today we'll continue talking about the transcription in eukaryotes. But before we continue talking about the transcription in eukaryotes, I want everyone to try to solve this question at home. So حاولوا تحلوا هذا السؤال. أنا متأكد إذا قدرتوا إنه تحلوا هذا السؤال بتكونوا فهمتوا أساس مهم جدا في الترانسكريبشن. So if you have the following RNA sequence, from 5' to 3'. So, what is the sequence in DNA? Try to write it down with the correct directions. So, إذا عندكم هاي الRNA بدي إياكم بس تحاولوا ترسموا الدبل ستراند that produce these RNA molecule. هذا السؤال لكم وحلوه لأنفسكم. Now, you remember that transcription in eukaryotes produced a pre-mRNA that contain introns and exons. And you remember that the introns are kind of RNA or kind of non-coding RNA that should be removed from the pre-mRNA to produce the mature transcript or the mature RNA or the messenger RNA. You remember also the exons are the code for the protein synthesis. So what we call the process of removing introns from exons, we call it RNA splicing. This is the mechanism how the introns removed from the pre-mRNA and keeping only the exons for protein production. حكينا إنه removing of introns from exons is mediated by a mechanism called RNA splicing. هلا خلينا نسأل السؤال التالي. The splicing of RNA, is it going in the same way all the time and producing one type of proteins? In fact, sometimes the RNA can be spliced in different ways to produce different mRNAs and therefore different proteins. And this mechanism called alternative splicing. So splicing the RNA in different ways to produce different proteins, we call it alternative splicing. The produced proteins called protein isoforms. Protein isoforms usually doing the same biological function among different cells. Let us take an example about the alternative splicing and important protein, which is the tropomyosin alpha. Tropomyosin alpha is a protein involved in muscle contraction, also playing different roles in different types of cells. Also, it is expressed in muscle cells and in brain cells, fibroblast cells, but in muscles it do muscle contraction or involved in muscle contraction. As you can see here, that in smooth muscles and striated muscles, we have two different isoforms of tropomyosin alpha. And the two protein isoforms actually produced by two alternative splicing. You see here, for example, in smooth muscle cells, the exons code the tropomyosin alpha, including 1, 2, 4, 5, 6, 8, 9, 10, and 14. But it is not the same for the protein isoform in the striated muscle cells. You see 1, 3, not the 2 like in smooth muscles, 4, 5, 6, 8, 9, 10, 11, and 12. Actually, in both cells, the tropomyosin and the 2 protein isoforms doing the same function, muscle contraction. But you can see that we have different code for each protein isoform, and the different code results from the alternative splicing. You see here that we have some exons are alternative, so they can be changed among the protein isoforms in green color here, as you can see. And some of the exons are constitutive exons. They are fixed. See, like the exon 1, 4, 5, 6. Here maybe you get confused because we have 8 and in green color. Do not confuse, because this exon, it is the same in smooth muscle cells and striated muscle cells, but it will be different in the other protein isoforms or other tropomyosin isoforms in different cells. So we added this in this exon 8 in green. Now, again, the alternative splicing can produce different mRNAs, and the different mRNAs produced different proteins called protein isoforms that do same biological function in different cells. You remember that I mentioned before that the transcription in eukaryotes is more complicated than in prokaryotes, and the transcription in eukaryotes is highly regulated, and there are a lot of processing for the mRNA in eukaryotes before it became ready for protein translation. So, what are these processes that occur to the mRNA in eukaryotes to prepare it for the protein translation? These processes including the capping, adding the cap at 5' end. We will talk about it in the next slide. Splicing. You remember we described the splicing mechanism, removing of introns and keeping the exons, and the polyadenylation, which is the addition of adenine monophosphate to the 3' end of the mRNA. For sure, all of these processes required proteins and enzymes, and some of these proteins associated with the phosphorylated tail of the RNA polymerase II. Remember that the carboxy terminus of the RNA polymerase, the carboxy terminus or the tail of the RNA polymerase II, is phosphorylated by the transcription factor 2H. Transcription factor 2H. Now, while the RNA synthesized, these proteins start jumping to the RNA from the RNA polymerase II to the RNA to start and do its function. For example, after synthesis the first nucleotides, the capping factors will jump to the RNA and start doing the capping. Also, for the polyadenylation, we have some factors and enzymes that also, once we reach the end of the RNA, it starts the polyadenylation through addition of adenine monophosphate to the tail of the mRNA. Now, let us talk about the addition of a cap to the RNA أو إضافة القبعة للRNA and how it occurs. Soon after the RNA polymerase II synthesized the first 25 nucleotides of the pre-mRNA, the 5' end of the pre-mRNA modified by a cap, and this cap consists of guanosine triphosphate. This modification performed in reverse orientation from 5' to 3' instead of the 5' or to 3' as the usual RNA or DNA synthesis. You remember that the synthesis of RNA or DNA is from 5' to the 3'. So maybe some of you confused in the directions. To make it easy to understand the directions 5' to 3' and the 3' to the 5', maybe simply you can take a look to the ribose sugar. It's a sugar. We have 5 carbons in this ribose sugar. So we call them carbon 1 from here, C1, carbon 2, C2, carbon 3, you see, carbon 3, this is where the 3' end, and carbon 4 and carbon 5, you see the carbon 5. That's why we call it 5' and 3'. It's where the linkage, the direction of the linkage occurs. So as you can see here, the addition of the GTP or the cap is in the reverse orientation from 5' to 5' to 5'. Carbon C5 and carbon C5 in the pre-mRNA and carbon C5 in the guanosine. So capping is not for nothing, for sure. It do some jobs for the mRNA. So what capping can do and what is the importance of the capping? Actually, capping can help to stabilize the mRNA. You remember that maybe I mentioned before that the mRNA half-life is short, and sometimes we need the mRNA stable enough to do the protein translation and all of these processes during transcription. So we need the mRNA to be stable enough for protein translation. So the capping can stabilize the mRNA. Also, it signals the 5' end of eukaryotic mRNAs. That helps the cell to distinguish the mRNAs from other types of RNA molecules which are uncapped. So it helps the cells to identify the mRNA from other uncapped RNA molecules inside the cells. Also, it helps to recruit proteins necessary for splicing and polyadenylation. Splicing, you remember, the removing introns from exons, and the polyadenylation, addition of adenine monophosphate to the tail of the mRNA. Also, it helps in exporting the RNA to the cytoplasm. You remember that the replication and the transcription occurs in the nucleus in eukaryotes, and the processing of the pre-mRNA into mature or messenger RNA should occur inside the nucleus. After maturating the RNA should be exported into the cytoplasm, where the translation occurs. So the translation occurs in the cytoplasm. So the capping or 5' capping can help to export the mRNA into the cytoplasm. Also, it helps in the translation of mRNAs to proteins. Now, we talked about the capping and splicing processes. Now let us talk about the third process, which is the polyadenylation. You remember that we mentioned in the termination phase in the transcription mechanism in eukaryotic cells that there is a signal that told the RNA polymerase to stop synthesis for the RNA. And this signal consists of a consensus sequence of a U, A in the mRNA, followed by a GU-rich region. Actually, this signal telling the RNA polymerase to stop the synthesis of RNA and terminate the synthesis. Now, let us describe this mechanism in more details and how it occurs. There is a signal or a sequence, in the mRNA, which is the AAU, AAA at the 3' end, can be recognized by an enzyme or endonuclease enzyme and cleave the pre-mRNA at the CA nucleotides at the 3' end. Actually, this step mediated by a endonuclease enzyme. So you remember that we have exonuclease and endonuclease enzymes. So what is the difference between endonuclease and exonuclease? Actually, the exonucleases usually cleaves the ends, but the endonuclease cleaves in the middle or inside the nucleic acid sequence. So from the name endo and the name exo. After cleaving the pre-mRNA, there is an enzyme called poly-A polymerase. The poly-A polymerase will start adding the adenine monophosphate nucleotides to the 3' end of the mRNA. And the poly-A polymerase can add around 200 poly-A monophosphate nucleotides to the 3' end of the mRNA. But does that require a template for the poly-A polymerase to add the A monophosphates to the 3' end? Actually, poly-A polymerase does not require a template, and the poly-A tail is not encoded in the genome. So the poly-A tail just added by the poly-A polymerase, so we don't need a template that required for the RNA, for the poly-A polymerase to do the poly-A tail. So we don't have the poly-A in our genome. Now, what is the significance of polyadenylation and the importance of polyadenylation? Actually, there are important functions for the addition of poly-A tail to the 3' end of the mRNA. The polyadenylation helps in transporting the mRNA from the nucleus to the cytosol. And also it helps in the translation, the production or the synthesis of proteins. Also it helps to stabilize the messenger RNA. Now, let us talk about the messenger RNA transport. You remember that the transcription occurs in the nucleus, and all of the processing for the pre-mRNA to mRNA occurs in the nucleus, and then we need to transport the mRNA from the nucleus to the cytoplasm where the protein translation occurs. Actually, this transport for the mRNA is highly selective and is associated with the correct RNA processing. What do we mean by that? We mean by that if there is any defective RNA molecules like the interrupted RNA molecules, mRNA with inaccurate splicing, and so on, will not be transported outside of the nucleus. So if we have any mistake or wrong processing in the RNA or interrupted synthesis or interrupted RNA, then there is no transport for this RNA to the nucleus. Why is that important? You know, imagine that if we have like RNA partially transcribed and processed, maybe we let us say that we have a gene with 10 exons to code a certain protein. If we have like mRNA with the first 5 exons, then imagine if it is transported into the nucleus, and maybe short proteins or incorrect proteins will be produced. So there is no that will affect the cell function. So our cells has the capability to control the transport of correct mRNA to the cytoplasm, allowing the synthesis of correct proteins. Now, let me ask the following question. What will happen to the mRNA after doing its job? They will stay forever? For sure, no. In previous lectures, maybe I remember that I told you that the significance of any mutation in the mRNA is not like the significance in the DNA. Because if there is any mutation in the mRNA, it will be eliminated because the mRNA will be degraded later on. But if there is any mutation occurs in the DNA, it will stay in our cells, in our DNA, and continue with our cells and our genome. So that's why the mutation is more significant in DNA compared to the mRNA. In both prokaryotes and eukaryotes, the mRNA will be degraded, but we have some little differences. In fact, in bacteria, for example, the majority of the RNA molecules will be degraded with a half-life of about 3 minutes. So as you can see that the RNA molecules in bacteria are very unstable. While in the eukaryotic cells, the half-life of the RNA is about 30 minutes and can reach 10 hours actually. You see that the RNA in eukaryotic cells are more stable than bacterial cells or prokaryotic cells. So, how is the degradation occurs for the mRNA? First, there is an enzyme called the adenylase enzyme, responsible for shortening and removing of adenine nucleotides from the 3' end. After that, the RNA will go either for the decapping and 5' to 3' exonuclease degradation, or it will go for the 3' to 5' exonuclease degradation. Now, let me ask the following question. What will happen to the mRNA after doing its job? They will stay forever? For sure, no. In previous lectures, maybe I remember that I told you that the significance of any mutation in the mRNA is not like the significance in the DNA. Because if there is any mutation in the mRNA, it will be eliminated because the mRNA will be degraded later on. But if there is any mutation occurs in the DNA, it will stay in our cells, in our DNA, and continue with our cells and our genome. So that's why the mutation is more significant in DNA compared to the mRNA. In both prokaryotes and eukaryotes, the mRNA will be degraded, but we have some little differences. In fact, in bacteria, for example, the majority of the RNA molecules will be degraded with a half-life of about 3 minutes. So as you can see that the RNA molecules in bacteria are very unstable. While in the eukaryotic cells, the half-life of the RNA is about 30 minutes and can reach 10 hours actually. You see that the RNA in eukaryotic cells are more stable than bacterial cells or prokaryotic cells. So, how is the degradation occurs for the mRNA? First, there is an enzyme called deadenylase enzyme, responsible for shortening and removing of adenine nucleotides from the 3' end. After that, the RNA will go either for the decapping and 5' to 3' exonuclease degradation, or it will go for the 3' to 5' exonuclease degradation. This is the end of today's lecture, and tomorrow there will be a short lecture. راح يكون في عندنا محاضرة قصيرة جدا، فيديو لمحاضرة قصيرة جدا, talking about some phenomena in eukaryotes. حقيقة المعلومات فيها قيمة جدا ومفيدة لكم, بتضيف معلومات مهمة جدا. So I will call it additional information lecture. غالبا هاي المحاضرة ما راح تكون داخلة في الامتحان النهائي. maximum راح يكون في عندي سؤال واحد فقط من المعلومات العامة في هاي المحاضرة. بفضل انكم تسمعوها او تقرأوها او both together, لانه فيها كثير معلومات مفيدة لكم. بس زي ما حكيت لكم غالبا ما راح تكون داخلة في الامتحان النهائي, واذا كان في